Our next speaker is Dr. Jordan Winter. Jordan received his undergraduate degree from Princeton and his medical degree from Cornell Medical College in New York. After completing a general surgery residency at Hopkins, he went on to MSK for his surgical oncology fellowship. Dr. Winter's research seeks to understand how pancreatic cancer cells survive in the tumor microenvironment and identify their metabolic weaknesses to ultimately discover new approaches to target pancreatic cancer cells. Today, he is going to tell us about a metabolic target that his lab found in pancreatic cancer. Jordan.
Okay, good morning. Well, it's a privilege to discuss with you some work that really has spanned the last decade in our lab, and I'm going to give you a broad, swift sweep of what has been, I think, a true bench-to-bedside effort that has culminated most recently in a paper this summer in Nature Cancer, and an opening of a clinical trial which we're actively accruing to. And all that said, I still think we're only on the 10-yard line with 90 yards to go in this endeavor. So, as Quentin said, our lab focuses on trying to understand the metabolic vulnerabilities in pancreatic cancer. It's one of the most aggressive cancers that we face, and part of the reason why it's so aggressive is that it's been able to adapt to a very stressful and austere microenvironment. But those adaptations, if we really understand them, can illuminate certain vulnerabilities and dependencies that we can then target, hopefully with a therapeutic window. This is the enzyme that we've become very interested in, IDH1. IDH1 is a metabolic enzyme. It's a homolog to an enzyme in the TCA cycle, in the Krebs cycle. But this enzyme exists in the cytoplasm, and this is the reaction that it catalyzes, where it converts isocitrate to alpha-ketoglutarate, and NADP is considered a cofactor, but we think of it really as a reactant, as a substrate that's every bit as important as isocitrate. The reaction goes in both directions, but under conditions in the cancer microenvironment, specifically pancreas cancer, the reaction proceeds predominantly to the right on your screen. So alpha-ketoglutarate is generated, NADPH are generated, and these molecules are both really, really important. Alpha-ketoglutarate, as this cartoon shows, feeds the TCA cycle. So even though isocitrate lives, or IDH1 lives in the cytoplasm, it really regulates the mitochondria by feeding it with this carbon substrate that allows the cycle to circle. And NADPH is the reductive currency of cancer cells. It primes all of the antioxidant defense, which is critical for cancer cell survival because the microenvironment is so oxidative and stressful, predominantly because of hypoxia and nutrient deprivation. The mitochondria are really important for cancer cells to survive because under low nutrient conditions, which is the case in these in the pancreatic cancer microenvironment, the cancer cell needs to optimize its ATP generation to survive. So we did a bunch of mechanistic studies showing that when we eliminate IDH1 from the system, all of these metabolic perturbations occur. And I've just shown one example here, one slide that's sort of getting into the nuts and bolts, the nitty-gritty of the metabolism consequences. We can label carbon and glucose and glutamine and feed it to cancer cells and actually measure the heavy carbon in all of the different compounds in core metabolic pathways. Those are those red circles that are labeled carbons. And when we do that, we can see that when we knock IDH1 out of the system, so we delete it with CRISPR, in the top left figure, you can see that the labeled carbon in alpha-ketoglutarate, which is the product of IDH1, is diminished, particularly at that fraction where 2 of the carbons are labeled. That's 1 spin of the citric acid cycle, where we see labeling is diminished in IDH1 knockout cells. But in the bottom right, you can see that all of the TCA metabolites are actually reduced when we knock out IDH1. So even though it's cytosolic, it has strong implications for mitochondrial function. So after we determined that there are all these metabolic and mechanistic consequences of ablating IDH1 genetically, we wanted to understand whether that translated into actual cancer cell survival. And indeed it does. And in just walked the postdoc Ali Vaziri Gohar, who's done 90% of this work. What Ali showed was that under low glucose conditions, where IDH1 is especially important, the knockout cells don't live. And this is a colony formation assay where you can see that under low glucose conditions, that's 2.5 millimolar, there's no cells that live. And that's the same whether we look at either of these cell lines where we were able to knock out IDH1. And when IDH1 does exist in the plus cells, of course the cells suffer somewhat under low glucose, but they still are able to exist, showing that IDH1 is indispensable under low nutrient conditions, which simulate the tumor microenvironment. We tried to simulate that exact experiment in animals, in mice, by driving up their blood sugar levels and increasing the glucose levels in the tumors to see if IDH1 was dispensable under higher glucose conditions. And what this xenograph study shows, so these curves measure tumor volume over time when we implanted them in the flanks of mice. You can see that the IDH1 knockout cells, which is the red curve, those tumors don't grow at all normally. But if we allowed the mice to consume sugar water and drive up their glucose levels, you can see that there's a 3-fold increase in the glucose levels in the tumors when they do that. All of a sudden, the cancer cells grow at a normal rate. So this experiment demonstrated that IDH1 is dispensable under high glucose conditions, but under normal physiologic conditions, it is indispensable. The tumors can't live without it. So now we know that the target is important. That's target discovery. How about figuring out how do we actually inhibit the target with a drug so that we can translate it into patients? And this was one of our most interesting findings, and that is that a drug already exists and we didn't even know it that targets wild-type IDH1. See, 1% of all cancers have a mutation in IDH1, and that mutation in those cancers, they occur in leukemia, glioma, some sarcomas, and bile duct cancers. That mutation seems to be cancer driving in those tumors. And there is a drug that targets that mutant enzyme. But what the manufacturers of the drug thought was that the drug was selective for the mutant enzyme and spares IDH1, the normal version, in normal cells. And indeed, they were correct in their simple bench laboratory assays where they looked at standard media. The drug only inhibited the mutant enzyme and not the wild-type enzyme, and that's because there are high magnesium levels in normal tissue culture media, as there is in serum, and the magnesium competes with the drug in the wild-type enzyme, but it doesn't successfully compete with the drug in the mutant enzyme. That has to do with the enzyme kinetics as it relates to the magnesium in a specific binding pocket. But tumors, we found, have much lower levels of magnesium, and when you reduce levels of magnesium, all of a sudden that drug becomes very potent against the wild-type enzyme. And in fact, we looked at all of the off-the-shelf IDH1 mutant inhibitors, drugs that were specifically designed to target mutant IDH1 cancers, and in every single one of these drugs, what we see is when we reduce the magnesium levels, and we did an IDH1 activity assay, every single one shows that the activity of wild-type IDH1 is reduced by these drugs, which previously were thought to be selective for the mutant enzyme. Ivosidenib is the most interesting because it's already FDA approved and is safe and orally available in patients, and it's approved to treat IDH1 mutant leukemia and bile duct cancer. We sought to apply it to wild-type IDH1 pancreas cancer since there are no IDH1 mutations in pancreatic cancer. So how does it fare in our preclinical models when we test ivosidenib in our pancreatic cancer models? Well, the first thing is we needed to determine whether these artificial conditions where we drop magnesium levels in cell culture were even relevant in cancer models. And indeed, you can see that in the pancreatic cancer xenograft, magnesium levels are only about 10% of the levels seen in serum. And that does jibe with just one study ever done looking at intratumoral magnesium levels performed 50 years ago in breast cancer, where magnesium levels were found to be just under half the levels in serum. We're currently looking at levels in pancreatic cancer in our patients. But you can see that for that reason, the drug is extremely effective against pancreatic cancer without wild-type, without IDH1 mutations. The tumor actually shrinks when we give the mice the same oral dose that was given in all of the drug development experiments against IDH1 mutant tumors. In fact, the drug works better against this pancreatic cancer model than it did in any of those experiments that led to the drug's development and approval. Even more impressive, we tested this in probably a dozen different models, but we asked a very well-known mouse modelist who's across the pond in the UK to do this experiment independently in the most aggressive pancreatic cancer model. It's a genetically engineered model where they naturally develop pancreatic cancer that looks just like human pancreatic cancer. And in that experiment, using the drug that we've been using, Dr. Morton found that there's an improvement in survival in that most aggressive pancreatic cancer model with this single agent drug, which is very hard to achieve. So as a result, we've taken this work and we've opened up a clinical trial in our patients here at university hospitals. It's a single institution study with 16 patients and patients with resectable pancreatic cancer. They're all going to get surgery and they are all going to get neoadjuvant chemotherapy, standard of care, plus ivosidenib for 12 weeks, 2 weeks of monotherapy followed by 6 weeks of combination with chemotherapy, followed by 4 weeks of monotherapy while they're recovering from their chemo, and they get the drug all the way until the day before surgery, and then we do their surgery and do all sorts of cool metabolism studies on the resected specimens. We've actually enrolled one patient into this trial. The drug was extremely safe. This is a phase 1 safety trial. She did very well with surgery. She went home in 5 days and is doing very well, and we were able to get all of her tissues to eventually study them. So that's the brief overview of this line of investigation. I'd like to thank my entire lab who have done all of this work, particularly Ali, who is finishing up 6 years in our lab with us and has just done a brilliant job, and happy to take any questions. Thank you. Ted.
Ted, can you wait for the microphone?
So great presentation, really exciting findings. So I guess a follow-up question is, under normal glycemic conditions it works. Is it even better under hypoglycemic conditions?
Hypoglycemic.
Yeah, if you're giving low glucose diets or more even ketogenic diets and those sorts of things.
Yeah, I mean we're very excited about that idea. And the best way to drive glucose levels down is through a ketogenic diet where only about 5% of the calories are glucose. And Omid Haji Hassani in our lab is a grad student who's doing those exact experiments where just by a ketogenic diet he can reduce the glucose levels in the serum by about half. That translates into lower glucose levels in the tumor. And as a result, it is very synergistic and making the drug better. We're very interested in trying to find the best therapeutic pairings for this drug, realizing that monotherapy is not going to be the answer in our patients. So ketogenic diet seems to be one exciting possibility.
it's impressive that this is an oral formulation. Do you think about intravenous formulation or anything like that would be better prospect, or is it best that it's oral because it is pancreatic and it's part of the way it's metabolized, gets to the pancreas better? Because I know there's problems with some other therapies being intravenous because of the microenvironment with the pancreas.
Well, I mean, that question, so the question was, is the oral formulation the best possible formulation, or might there be better tumor penetration intravenously? I don't know the answer to that question. We've given it intraperitoneally into the mice, and we can use a lower dose, and the results seem to be the same. I think my general sense, and I'm not a medical oncologist, I'm a surgical oncologist, but my sense is that if a drug is orally bioavailable, that generally is seen as a very good thing because it's just easier for patients to take, it's easier to administer, it's more cost effective. And so we're very excited by the fact that it's orally administered, and one of our compelling pairings that we're looking on developing is with another oral agent to have a totally oral regimen that attack the drug in two different ways.
Jordan, obviously your lab has a focus on pancreatic cancer, but you made a pretty strong case, especially with the preliminary or the prior data in breast cancer, that there's a similar microenvironment problem of low glucose and low magnesium. So it sounds like you could go to a lot of other cancers. What's your list after pancreas, colon, breast?
Yeah, so as I said, that IDH1 mutant tumors are 1%. We think this drug works in the other 99%. And so far, every single tumor that our lab has tested, and everybody in our lab has done experiments with this drug in mice so far. And that list, just in our lab alone, has included melanoma, colon, ovarian, lung cancer. So any wild-type tumor seems to be vascularized in the center of these tumors, and we think that the drug should have activity.
So, Joe, quick question for you as well. Bench to bedside, taking a laboratory finding from the bench to the clinical space is often a very big lift for cancer investigators. Can you tell us the process that you took to take your discovery to the clinical trial? And also, how did you get buy-in from the industry partner?
Yeah. The bench to the bedside step is a totally different skill set and a totally different effort. You could be a genius in the lab and never translate your brilliant discoveries to the clinic because you don't have the right clinical partners or you don't have the right buy-in from your clinical collaborators. It has been such an enormous effort just to create this trial. Obviously, writing the protocol, but also making sure that I had a really reliable and invested medical oncology colleague, and trying to figure out how to secure funding, which is totally different, securing basic science bench funding from securing clinical trial funding. So in this case, we secured funding from a cancer foundation called Gateway for Cancer Research, and they contributed $1 million to this trial. We also have philanthropy. John and Peggy Garson, who are the benefactors to my endowed chair, contributed $500,000, and we think we can do the trial for $1.5 million. The industry partner, in theory, would be Servier Pharmaceuticals, which currently owns the asset ivosidenib. They purchased it from Agios, and believe it or not, they are not fully engaged at this point. They have other priorities, and so we think that taking it to the next steps really would be important to have them engaged and trying to figure out every angle, back door, front door, side door, underneath, to get somebody in that company to get excited about this has been just an endless pursuit of ours and something that I have not given up on. I'm hoping with each new return of data, they might get a little bit more intrigued. But we've even engaged people on their board. But short of that, I think that the most practical way in some ways, unfortunately, in some ways it might be fortunate for us to really go forward, is to develop our own compound and own our own compound. So we finally said we need to go full forward with that effort, and Drew Adams is helping our lab screen new molecules, and will do hit to lead to try and develop our own asset, and then we, I think, control our own destiny better if we can be successful in that effort.